Hello, welcome to another video from the conscientious biologist, Ben Gallagher. This video is the second in a sequence of 3 on transport through membranes. This one focuses on osmosis, and I very strongly suggest you've watched the first of the transport through membranes videos, which was on diffusion and active transport. This video is suitable for GCSE level, and please could I ask that if you enjoy what you see, you subscribe to my channel to get updates on future uploads. Thank you. So let's start off by asking the question, what is osmosis? How does it fit into this whole transport through membranes section of the course? Now, it's a little bit of a cheat because when we say osmosis, all we are talking about is the diffusion of water. It's one of those weird little quirks that at some point in time, someone decided that water should have its own name for its diffusion. The diffusion of carbon dioxide is just called diffusion of carbon dioxide. Diffusion of nitrogen is called diffusion of nitrogen. But for some reason, the diffusion of water gets its own name, that name being osmosis. However, there is one key little difference between osmosis and the diffusion of water. And it's one little stipulation in the definition of osmosis. So let's look at that first. Osmosis is the passive movement of water, passive meaning it doesn't require energy, from an area of high water concentration to an area of low water concentration, this is the key bit, through a semi-permeable membrane or partially permeable membrane if you're happier with that term. Now what that means is it must be through a membrane, either in or out, mostly we're talking about of a cell. If you imagine when you boil your kettle and you see the steam coming out of the top of the kettle, that steam is water molecules moving from an area of high water concentration in the kettle out into the air where there's a lower water concentration. But because that isn't through a membrane, that is just diffusion of water when you see the steam rising and spreading out into the air. It must be through a membrane if it's going to be classed as as osmosis, which means osmosis is essentially just a biological thing because it's generally only biological systems that have membranes. Before we carry on, let's just dissect this concept of water concentration, because this is one area where students often struggle because you have to flip your thinking around a little bit. So imagine I've got 2 glasses. Let's call them glass A and glass B. Into glass A, I put a certain amount of orange concentrated cordial or orange squash, whatever you call it. In glass B, I put a smaller amount of that concentrated orange, and then I top both glasses up to the same level. You can look at them on my diagram there. They look slightly different colours. Glass A is more concentrated. It's darker in colour because it's quite clearly got more bits of orange in it. So if I asked you which glass is the most concentrated, pretty much all of you would go for A. Any logical person would. Which would be true. If we were talking about the orange, we could apply 2 key terms to that. That glass A is concentrated. There's lots of orange particles. Glass B is dilute. There's very few orange particles. But if we're talking about osmosis, then we're talking about the water concentration. And glass B has a higher concentration of water particles than glass A. It has less orange, which means it has more water. It's about the comparison between the two. So essentially what we're talking about here with osmosis, and we're talking about the water concentration and the stuff dissolved in the water, we have to go back to your sort of year 7, year 8 stuff, and we have to think about this solution, the concept of solutions. And a solution is when you've got a solvent, which is a liquid, and a solute that you dissolve into it. In our example here, we've got the solute being the orange particles dissolved in the solvent, which is water. Now, when we're talking about osmosis, we're only interested in water. So if you look at the little equation there, let's just switch out solvent for water. So when we're talking about solutions, we're talking about water with solutes in. And the more solute you've got, the lower the water concentration. The less solute, the higher the water concentration. Hopefully you fully understand now what we're talking about when we say water concentration. So let's put that knowledge into action now, and let's look at a key example. If I pull a plant cell up onto the board there, you can see that on that diagram I've got all the internal structures that are inside a cell, chloroplast, nucleus, all that kind of stuff. we're not going to worry about that when we're talking about osmosis, because we're only talking about the water that moves through the cell membrane. So let's just get rid of that to start with. Let's bring in a second cell now and put them side by side. Now, there's going to be a solution of water in the cytoplasm of one cell and a solution in the other cell. Again, like the glasses, let's call them A and B. And let's say that cell A has got 9% solute, that's the non-water part, in its cytoplasm. And cell B has 6% solute in its cytoplasm. So I've given you the non-water parts, the dissolved stuff in the cells. Now, if you think, osmosis can happen where the membranes are touching, so basically just inside this yellow box that I've put on there. But where the cells are touching each other, water should move from one to the other. So just take a little second, pause the video if you need to, and have a think, which way is water going to move? Is it going to move from cell A into cell B, or is it going to move from cell B into cell A? Have a little think, pause the video if you need longer. Okay, let's go through that then. The water should move from where it's at its highest concentration to where it's at its lowest concentration. So if you're still not sure, let's give you a little hint there. If cell A was 9% solute, it must be 91% water, because that gives you your 100%. Cell B must be 94% water, because the 94% water with the 6% solute again gives you your 100%. So hopefully now you can see cell B has a higher concentration of water than cell A. So osmosis is going to happen across the cells in that direction. Let's increase the complexity now and look at a whole cluster of plant cells. Now, don't be at all intimidated by that. The theory is exactly the same as we looked at on the last slide. The only difference now is we've got 5 cells there on the screen, and we're going to look at how water moves in between all of those. So let's give you the solute concentrations again in these. And again, I've labelled them A, B, C, D, and E. A has got 8% solute in there. So just try and think, if it's 8% solute, that's 8% dissolved stuff. The remainder must be water. So let's give you that one. That one's got to be 92% water. But have a little look around the rest of those cells. What I'd like you to do now is pause the video for a minute, however long you need, and try and think between all of the cells, A to B, B to D, D to E, any of them, which direction would the arrows go if you were marking osmosis? So pause the video now and have a think about that. I'm going to hope that you paused it and had a go. If you didn't, I'll carry on anyway and reveal a few more details that might help you out on it. So let's throw up there the water concentrations. I gave you the first one was 92, but let's put them all on there now. That should make it much, much more obvious. Again, now you've got that information. Pause the video again if you didn't have a go last time and see if you can still do it. You may or may not have paused it again. I'm going to carry on anyway. So looking at those, we should be able to start putting the arrows in to show the directions that it moved. So let's go between A and B first. Does it go from A to B or B to A? Well, 92% water in A is clearly higher than the 88 in B, so it's going to move in that direction. From B to C, the 93% in C is higher than in B, so it's going to go back that way. Let's go C to D. 95 is higher than 93, so it's going that way. And D to E, have a quick think if you didn't before, it's going down, the 95 to the 94. Hopefully you got all those arrows correct, because that's quite a straightforward way of checking your understanding of osmosis. Of course, we've done A to B, B to C, C to D, and D to E. Any membranes that are touching though will do osmosis. So you've also got osmosis on those arrows that I've just added. Anytime there's 2 membranes touching, water will start to move through the 2 cells into each other. Now, all the examples so far, we've used plant cells. There's a reason for that, which we'll explain in a minute. But let's just check on osmosis in animal cells. So I've got an animal cell in the middle of the screen there. Again, like the plant cells before, you've got the nucleus, you've got the mitochondria there, the little red dots are the ribosomes. We're not going to worry about those because we're only worrying about the water that can move in and out through that outer cell membrane. But let's imagine initially we've got this animal cell and we've put it under a condition where it's going to lose water. Now it's only going to lose water if it's in a solution where the solution around the outside of the cell has a lower water concentration. So there's more water in the cytoplasm of that cell than there is in the solution outside of it. That means it's going to lose water. So if you look to the cell over on the far side over there, you can see that it's shriveled down a bit. It's still going to be able to function, but some of its metabolism may be inhibited if it needed the water to do any kind of breakdown processes. If we put that cell into a higher concentration of water, the water is going to move back into the cell and restore it to how it was. Now let's think about what would happen if we took that cell in the middle and we put that into a concentration of water that had a higher concentration than the cytoplasm in that cell. So if we took that cell and put it into pure water, let's say. In pure water, water is going to move from the pure water into the cell. But of course, cells, like balloons, can only increase and swell up a certain amount. So what would happen is that animal cell would just burst, would rupture, and would therefore no longer be a functional cell. So osmosis in animal cells is a little bit more delicate and can have massive consequences. If water moves into your cells, they could swell up, they could burst. So putting animal cells in high concentrations of water is rarely a good idea. Now let's take exactly the same principle that we've just described with animal cells and apply it now to plant cells. So in the middle of the screen there, I've got a plant cell. Now, same as before, if we put it under conditions whereby it lost water, so we put it in a solution that had a lower water concentration than the cytoplasm, it's going to lose water, and you can see exactly like in the animal cell, the cell membrane, that's the blue line, has shriveled in and pulled away from the cell wall. Now, if you think back to the cell biology lessons, we talked about the cell wall, we talked about how it's made of cellulose, so it's a relatively strong, rigid structure. That's not going to be pulled in when the cell shrivels. Now, when the cell shrivels in like that, though, it makes the entire structure unstable and floppy. Now, the word for that is flaccid, but we can describe the cell, because its membrane's been pulled in away from the membrane, we say that the cell has plasmolized, and it's become flaccid, which means the whole cell is now more floppy, less stable. It's not as solid as it was. If we put that cell back into concentrated water, it's going to go back to its original state. But if it was in very high concentration water, but now think about what happened to the animal cell. The animal cell burst, but in a plant cell, the membrane is inside a box, inside the box of the cellulose cell wall. So that membrane has got to push itself out against the wall. But it can't burst because it can't increase beyond the cell wall. So what happens instead of bursting is the plant cell will become pressurized. Now think about the tires on your bike or the tires on a car. When you pressurize them, you put extra air in them, they become really hard. They become solid structures. Now this is a really, really good thing for the plant, because unlike us, plants don't have skeletons to hang all their cells off and hold them upright. So they rely on their cells becoming pressurized and solid so that each cell can act like a brick that you can build up on, much like a house that's built out of bricks. A house doesn't really have a skeleton, it's just got the bricks that are strong in their own right. Each plant cell must be strong in its own right. And that pressurized form of plant cell is said to be turgid. That means it's pressurized. Now, when a plant's cells are all turgid, the plant can be upright and solid like a structure like the house that we've just mentioned. Whereas if its cells are all flaccid like the ones over on the far side there, that's when the plant has to bend down and it wilts because its cells are no longer strong enough or solid enough, because they're not pressurized, to hold everything upright. They've gone flaccid, they've gone floppy, they all bend over. Now, of course, a pressurized turgid cell could lose water, could start going across in the other direction, but generally with plant cells, they're either turgid or flaccid. Even that one in the middle would be turgid enough to keep the plant upright. It's only when the cells become totally flaccid and plasmolized that the entire plant would wilt. So what I'd like us to do now is take that knowledge of turgid and flaccid cells and go back to the diagram that we had a couple of slides ago, this one here that's on the screen next to me. So we established the direction that the osmosis would happen between all of these cells. But now I want us to try and apply that knowledge from the last slide and decide which one of these cells would become turgid, which ones would become flaccid. Now that's not as easy as it sounds, because you need to think back to the stuff that we talked about in the diffusion presentation, the first transport through membranes presentation, and we need to consider the concentration gradients. So let's look at cell A to start with, the one over on the furthest side there. It's 8% solute, it's 92% water. Well, you can see the arrows on there. It's gaining water from cell E and cell D, and it's losing to cell B. So you might automatically think, well, it's gaining twice, it's losing once, therefore it's gaining more than it's losing. If it's gaining more, it's going to increase in water, increase pressurized, and it's going to become turgid. Now let's just check that though by looking at the concentration gradients. Look at the arrow that goes between cell A and cell B. Now cell A was 92% water, cell B was 88. The difference between those is 4. So if you look on the arrow, I've now put a 4. That is the difference between 92 and 88. So that represents the concentration gradient there being 4. If you look at the other two arrows, both going into cell A, you can see the first one has a concentration gradient of 3 because it's going from 95 to 92. The difference is 3. The third arrow there is 2 because it's going from the 94% water in E to the 92% water there. So now that we know the concentration gradients, we can work out what the overall osmosis figure will be, because we've got the 3 and the 2 coming in. So if we have 2 plus 3, that gives us 5. But we're losing at a rate of 4. So we have to take that 4 away. So the 2 plus the 3, that's what's coming in, minus the 4 that's going out, gives us an overall figure of plus 1. There's one more unit of water coming in than is going out. So it is gaining more. It's still plus, it's up on what it was. So it's going to become turgid. So you can see we can put a T in there, T representing turgid. The key is there above my head. So that one's going to become turgid. Pause the video again there. Have a look at those cells. Have a look at how we calculated those different figures and see if you can work out which ones are turgid and which ones are flaccid. Please do have a go at this because it's a real key skill. So pause the video now. Did you cheat? Are you just waiting for me to talk? Hopefully you did pause and have a go. So let's have a little look here and let's start filling some of these in. I want to jump straight across to cell C, that one that's just up there, because C is quite an interesting one. Because look at the arrows here. It's got 2 arrows coming in, one from E, one from D. It's got one going out, going into B. Its arrows are exactly the same as cell A. So you could be forgiven for thinking, cell C must become turgid because that's what A did and it's got the same number of arrows. But remember, it's about the rate of osmosis, okay? Just like when in the diffusion presentation, it's about the rate. So let's look at those 3 arrows and put the rates on. Now you can see that arrow at the bottom there, at the bottom of cell C, coming from E, is only 1 unit. The concentration gradient is only 1. So that's a tiny flow, a dribble of water coming into it from cell E. From cell D, the figure is only 2. Again, that's still only a little dribble of water coming in. Whereas, look at the water that's being lost from cell C to cell B. That's 5. The concentration gradient is 5. So now if you consider it there, you've got your 1 plus your 2, that's 3 units coming in, but 5 going out. So overall, you should have worked out that that's minus 2. So even though you've got 2 arrows coming in and only 1 arrow going out, the outflow is still greater than the combined inflow from the other 2. We've still got minus 2. So if it's losing water overall, that one's becoming flaccid. Okay? Just in case you got any of them wrong before when you paused it, hopefully, and had a think, pause it again and just have another check. Can you do B, D, and E yourself? Pause it now. Hopefully you've done that, so let's carry on. And you should hopefully have worked out that B is taking in a massive amount of water. All 3 arrows are going into B, so it's clearly going to become very, very turgid. D is losing a lot. It's got 4 arrows going out of it. That's not surprising because cell D has the highest water concentration of all the other cells, so it's going to be losing to all other cells. It's -13, it's losing a lot of water, so that's going to become very, very flaccid. Cell D is going to go very floppy. E is losing water to A, it's losing water to C, it's gaining from D. None of its 3 arrows are particularly strong, 2, 1 and 1, but still when you work it all out overall, that's a -2 figure, which means it's going to go flaccid because it's still losing. So very, very quickly to finish up, let's just recap on what is osmosis, what you should have learned through this presentation, and that it is just the diffusion of water, but its key definition ends with that it must be through a semi-permeable membrane. If it's movement of water not through a membrane, then it's not going to be osmosis. Go back through and review this presentation because there's a lot of complex information here and a lot of tasks that you've got to do. There's some key details in this presentation that I've left out that will be in the next presentation. They're in the examples of osmosis. That's the Transport Through Membranes 3 lesson. Please do go and watch that one because that's got details that I left out of this one because I didn't want to make this one too complex. So once you've watched this one, go and have a look at that, which looks like this. So you can see it's Transport Through Membranes 3, examples of osmosis, and that's for GCSE. And as I said at the start, if you have enjoyed this, if you do find them useful, then please do subscribe to my channel. It really does help me out the more subscribers that I've got. Thanks for watching.